Welcome to Biophysics Day by PhysU, Ursa, and Biophysics TO. We hope you guys enjoy our event. First up, we will have bullet talks, starting with Professor Ryu.
All right, I have this. Yeah. Okay. Hello, everyone. I'm Will Ryu. I'm in the physics department and at the Donnelly Center. And basically, I'm interested in how biological systems process information, how they make sensory measurements, how they encode these measurements, and how they produce adaptive behavior. And I study C. elegans. And the reason C. elegans is a good organism for studies like this is that it really reveals the complexity of sort of this black box. Right? The system is just complex enough to give interesting behavior, but it is also simple enough to be tractable. So what I mean is that C. elegans has about 1,000 cells, 300 of them are neurons, and all these cells can be identified uniquely in each animal. Right? Also, the connectivity of the neurons is known. And so we can go and study a specific cell or specific neuron in one organism and study the same cell in another organism. Okay? Also, since it's a model organism in biology, we have lots of tools to perturb the system genetically. And we can do things like measure neuronal activity from all the neurons. And so here's a strain that we made that expresses a genetically encoded indicator of calcium in every neuron in the animal. And we're applying thermal signals to the system. And the fluctuations in the fluorescence are proxies for neuronal activity. All right. So what do I mean by behavior in C. elegans? So this is sort of a map of behavior. So we have the quality of information on one axis, and we have behavior on the left here, going from stochastic to deterministic behavior. So if you come into like this room, say, and all of a sudden you had no information, say it went black, okay? So what we're looking at is someplace down here. You would have some strategy to find some information somehow. Like you would say, you're in here, you can't see anything. You might do like a local search because your worldview is light switches are somewhere near the door, okay? And you do a local search. And all of a sudden you can't find anything. And then you would change the statistical strategy of your search. You might say, oh, I need to go to the front of the room now. And I would lengthen maybe like this random search by extending how long I move forward in the dark. Okay? If you came into the room and it was cold on one side and warm on the other side, what you would do is you would sort of randomly move around, right? And say, oh, this is where I like the temperature, okay? And maybe you choose a seat, okay? And that's because the quality of the information is a little bit higher, obviously, than having no information. And you can use that, and your behavior might be this balance of deterministic and stochastic processes. Right? And for example, now if you touch a hot stove, you're going to do a reflexive movement. You're going to try to escape from the stimuli because the quality of the stimulus has a lot of information, right? You're going to damage your hand. But there's still shades of that behavior, meaning it depends on how hot the stove is. And what basically modifies all of this, what modifies all of this behavior is the fact that these systems can learn and have memory. All right? So C. elegans sort of spans all these behaviors, okay?
I will extend your speaking time.
No, take away from this. Okay, so what I want to do basically is try to quickly give 3 examples from this map of behavior and give you a flavor of the types of experiments we do in the lab, okay? So we're going to be broad, not deep. All right. So physics of search. So if you look at C. elegans on a plate with no food, you can see the statistical strategy. It basically, say, starts at T0. Its life was good. It had food. And all of a sudden now, you can see this local search. It's looking very on a small length scale or time scale, right? But then there's no food. So just like that light example of you in the dark, you want to extend basically your forward movement state so that the search scale increases its time or length, okay? And I'm interested in understanding how C. elegans generates these statistics. How does it generate these random numbers, and how does it modify the distributions, right? It draws these random numbers from to determine its strategy, okay? So how one does this, we just look at C. elegans moving, right? And the movement and behavior is stereotyped. It's discrete. You can break it down into, say, states that are forward movements, maybe backwards, maybe turns, right? And for this talk, how long does it spend in time in the forward state? Okay, what are the statistics of the dwell time in that state? So to study this, we build these little gizmos, this desktop experiment, and we can see the worms trying to search for information, but we have a much better scale, much better image of the worm as it tries to find something. Okay. And this suggests a coordinate system, right? A measurements of the behavioral output that we can use. And if you think about this, this is sort of like a traveling wave, right? And basically for the traveling wave, this gives us a framework to think about how to measure its forward movement, its backward movement, right? We just look at the phase of this traveling wave and then take the derivative of that, we get
swap. Yes. Yes. And you can tell me.
Something is wrong.
Yes. Oh, sorry. I'm sorry.
Okay. And so what we can do is just by looking at the worms moving around, we can, for every phase, phi, and every omega, we just calculate the acceleration of the worms as they move. We just do this across hundreds of worms. And we average that acceleration, and that's what's being shown here. Okay? The noise is modeled as the standard deviation around that mean. And that's what's being shown here. Okay? So now what we can do is I can tell you what the worm will do if these models are correct. What I can do is I can start with some phi and omega, and this landscape will tell me how to iterate in the future. It'll tell me how omega and phi should change. Right? And so that's what's being shown here. In this phase plot of phi and omega, if I start somewhere and I just allow the system to evolve dynamically, what we'll get are limit cycles in stable states. So the limit cycles here are positive omega and negative omega. These are the stereotyped movement states of the worm. They're forward movement and backward movement. Okay? And when you start the system somewhere and you look just at this deterministic part of the landscape, you'll end up either paused or in forward or reverse movement. So that's really interesting because it defines behavior, right? Just by looking at this landscape. But I'm not interested in that. I want to know the statistics of leaving the forward state. To do that, I have to put in the noise. And again, this noise also comes from the data. If I start in the forward state and I add the noise, then it will leave the forward state if the noise level is big enough. And if I want to investigate if this is real, meaning does it mimic the real worm, I can generate all these forward state events by simulation. I just start here, and I measure the time it takes for it to leave that state. I make a histogram. This is the distribution. It looks fairly exponential. It has a tau of 16 seconds. 16 seconds is quite long. If we look at the real data and just measure the worm time in its forward state, you get something very similar. So there's some, authenticity to this, perhaps, right? It can recreate some parts of the movement. But what's more important is that it generates some framework of thinking about the problem. Remember, this is a complex problem. There's a bunch of neurons. Somehow it generates these random numbers, is evolved, gives you some strategic, probabilistic distribution so that I can find information, right? We know that it changes this time. Early on, it's local. Tau might be 5 seconds. Later on, tau gets extended, so it searches a longer distance. How does the system change that distribution? How does it extend the tau? So this gives us a way to think about it. This looks like thermal escape, right? So now it's like a ball in a potential. How can we change the time the ball stays in this state? Well, what we can do is we could change the noise level, right? Or we can change the structure of the landscape. So we looked at that. We looked at worms later in time when we know tau is actually much longer. What happens is that it's not the landscape that changes. It's the noise level that goes down. Right? And then there's this idea, what is the landscape? Right? What is this forced landscape? Well, it's due to the complexity of the neurons, the connectivity. Right? So somehow this complexity we can think of in a lower dimensional sort of manifold, a lower dimensional landscape, a simplification that's due, though, to a higher dimensional connectivity network, right, in principle. So we should be able to change the landscape. Because if I go in and change the neuronal network or look at a mutant that has some biochemical signaling that's pervasively defective, we should be able to change the landscape. And we do that in this paper. We look at mutants that are defective broadly, and we show that the landscape can be modified. Okay, so that's experiment 1. Experiment 2, physics of learning. So another very interesting thing about the system is that C. elegans can learn. Right? So if you take C. elegans and you put it with some temperature and food, it will remember that temperature. So it takes this analog value of temperature, right, and stores it somewhere. Okay? This memory is plastic. If I move that worm to a different temperature with food, it will forget the old temperature and encode the new temperature. Okay? So how do we investigate this? How do we investigate this memory and this learning? We look at behavior. So I can tell what the memory is for a worm by looking at thermotaxis. If I put worms on plates, Petri plates, and put it on a linear gradient, the migration, the taxis of the worms will indicate that it wants to go to a specific temperature. So we can investigate this by just taking video. Here it's sped up. This is the Petri plate. The worm's memory is over here. It wants to go over here. I want to measure the dynamics of memory. I want to quantify it. I want to model it. Okay? So to do this, we want basically this dynamics. This takes a long time to do. So what we've done is we took this Petri plate and we shrunk it down into a little microfluidic well. And now what we can see is many single worms in these wells, right? And as the worm crawls around, it will tell us its thermal memory. It will tell us its preference. So if I grow up worms that are in the cold with food, and I interrogate them around 20 degrees on a gradient, the worms on average will go to the cold side, and vice versa. So here what we're looking at is the worms moving around, telling us what temperature memory it has. Here is an average of many of these worms. We turn on the gradient. Worms that were grown on the warm side end up over here. Worms that initially grown on the cold side end up here. And this is basically telling us something about the memory. This is not that interesting over 60 minutes. It looks like the memory just persists, right? But if we look at this over 4 hours, all of a sudden we see dynamics in the memory. Okay? And so what we see here is that evidence of forgetting. So we grow worms who want to go to the warm side, the red. We interrogate them in this weird little microfluidic assay. The system, the worm system, of course, doesn't stop learning. It continues to try to learn, right? So now the worm is in this little droplet. There's no food. And it's swimming in between these little variations in temperature. It's sampling the temperature, and nowhere is there food. The system continues to learn. So you might think, oh, I'm going to forget the warm temperature, right? And by looking at these dynamics, you'll see that it does forget. But it's not just forgetting. Because what happens is it just doesn't go to 0. It actually crosses over. It wants to go to the cold side now. It wants to go down to the negative side. And even more weird, it will oscillate and recover. This is seen in the psychology of learning. This extinguishing of memory and this recovery of memory. And we see it even in the simple C. elegans. Yes? Okay. Do you want to ask a question? You only have 1 minute. Isn't there a question session after? Yes. Okay. Good. Okay, so we want to model this. So what's interesting now is that we can model this, but the challenge is that we want to draw from different disciplines. Right? So of course, we have biology. We can look at mutants. This tells us something. We make defects in how it measures temperature, defects in how food is integrated, information is integrated. But we also have to go to the psychology of learning. That place too also has quantitative models. Right? What's an important model is this Rescorla-Wagner model. It talks about conditioning, like Pavlov's dog. But that model is one-dimensional. Can a one-dimensional model oscillate? And as a physicist, you think, no, it cannot oscillate. We see oscillation. There must be more than one. So now we think about the model or the modeling as physicists as a dynamical system. And there's ways to look at sort of delayed embedding, to look at the dimensionality. And this gives us more power. We bring those three things together, and we can come up with a model, right? I'm skipping a lot here. This model has a lot of parameters. But how do we test this model? Now that we have the model, we can do more experiments, change these conditions, have various responses for cold and warm worms. And what happens is that we can still fit all of the data globally, right? So this suggests that there's some, rightness about the model. Okay. All right. I'm going to take two or three more minutes. Right? I'm going to talk about the last project, physics of pain. Okay. Pain and physics. You guys know all about pain of physics, right? I'm talking about physics of pain, right? So I want to have a model of pain. Okay? And I want to do this with C. elegans. And the reason why you want the model of pain is because if I zap the worm will respond in a way that we can correlate how painful the stimulus is with its behavior. Why do we want to do that? You cannot biopsy for pain. I cannot biopsy you and tell you how much pain you're in quantitatively. If I want to understand the effects of genes, genetics, pharmacology on how it changes the perception of pain, then I need something quantitative. This is what we want to do here. So how do we do this? Again, we make a little gizmo. This gizmo tracks worms and it can apply a laser stimulus. Here I'm showing us melting agar, right, in little dots. Now I can track the worm, I can zap the worm on its head while it's moving, it's behaving. And from this response, I can now correlate the laser intensity or heat with its trying to escape. This is you touching the hot stove, okay? This is the response, it's trying to escape. This is your reflexive move from the hot stove. We average a lot of worms. This is what we measure. We correlate now basically the strong, medium, and weak laser intensity with this escape behavior. Lots of ways to look at this data, okay? I'll tell you one way. One way we looked at this data is we used an algorithm, an inference algorithm called Sir Isaac. What it does is it takes any data, okay, and will model it, basically, dynamically. Any data, okay? So the feat of fitting a curve is not that important. It will do almost everything, okay? How it does this is it ratchets up the complexity of differential equations to fit the data. It's penalized for being too complex. So it tries to be parsimonious, okay? So we do this. There's this input, laser, and then there's this observable. It can get more complicated with numbers of hidden variables, and they can be coupled together, right? So we run Sir Isaac. We get a model. It can fit the data, no surprise. There's some terms that were small. We remove them because it makes things simpler. And we get our system that can model this data. But what's important is not the curve fit. What's important is that this is interpretable, okay? We can see how these are connected. We can make a map of what's happening here, right? Each term has its place, okay? And what's more surprising is that many of these terms have equivalents in neurons that have been identified with the exact same function. Not every term, but many of these terms. Okay, so that's my time. I'm going to stop there. I'm just going to say these are people who did the research, and thank you guys.
We'll do questions at the very end, like at the panel.
Yes.
Okay, I can do one or the other.
Both. You need both.
No, I know, but I can do one or the other.
Oh, I see.
And hold on. Good. Okay. This goes in my pocket.
No, that could be that should be on. Both of them are microphones. One's for the room, one's for the video. Thank you.
Thank you. Okay, is that working? Yes, it is working. Okay, so I guess I'd like to start by thanking the organizers for the invitation and for this opportunity. I wanted to say a couple of things. First of all, I think you guys did a really good job at selecting 3 diverse male. There's also many female that are really, really great at quantitative physics and biophysics. And you know, no, I'm not scolding you. I'm just saying I see in the audience that there's a mix of XX and XY chromosomes, and I think that's fantastic. Okay. The second thing that I wanted to ask is, how many of you are physics students? Okay, well, I have something to confess, which is that I'm not a physicist. I'm an engineer, so I apologize for existing, but you know, you have to deal with us. Okay, so what I wanted to do today, Will said he wasn't gonna go deep. I'm actually not even going to try to go broad. I'm gonna be very shallow and narrow, and I'm gonna give you a very general idea of the work that we do in the lab, trying to understand how cells coordinate their behaviors and specifically their movements. Now, in my opinion, the best way to appreciate the importance of collective cell behaviors is to look at a developing embryo. And if you do that, you will find, for example, groups of cells that collectively constrict their surface to generate backlink forces that drive the folding of a tissue, and that's depicted here during gastrulation in a fly embryo, so the process that segregates the 3 germ layers. But a similar set of collective constrictions contribute to the initial folding of our neural plate to form the neural tube. And if those constrictions are not properly coordinated, you can get devastating developmental disorders such as spina bifida. Okay, if you keep looking at embryos, you will also find groups of cells, oops, sorry, I went too far. You will find groups of cells that collectively rearrange their positions to alter the shape of a tissue. And that is depicted here with another example from the fly embryo, and hopefully you start noticing a theme with the examples that I'm showing. So this is the process of axis elongation, which determines what's head and what's bum, and lays down the basic body plan. And again, this is not exclusive to that process, so similar coordinated cellular rearrangements contribute to the equivalent process in us vertebrates, which is the elongation of a neural tube, but also generally to any developmental processes where tissues elongate. So for example, our limbs extend from the original buds where limb progenitors are specified, and that process is in part mediated by coordinated cellular rearrangements such as those depicted here. And once again, if those cell rearrangements are not properly coordinated, you can get a range of developmental disorders from stunted limb development to anencephaly in the most extreme cases of neural tube elongation defects. Okay, so these are beautiful examples of cells acting together to drive embryonic development. Unfortunately, sometimes cells coordinate their behaviors to spread disease. And what's the most infamous example of that? What? Cancer metastasis, exactly. So we know, and it's been increasingly recognized, that metastatic cells, particularly from solid tumors, form these aggregates where you can find anywhere between 8 and 30 cells. These are metastatic skin cancer cells. And the cells within these aggregates coordinate their movement to invade efficiently. Now, we know that if we disrupt the mechanisms that these cells are using to move collectively, the cells may still adhere to each other within that cluster of cells, but now some cells try to move in one direction, some cells try to move in the opposite direction, and the invasiveness of this cluster is dramatically suppressed. So our hope is that if we can understand what are the fundamental mechanisms by which cells coordinate their movements, one may be able to intervene when collective cell behaviors should be happening and they don't, which is often the case in congenital disorders. Or you should be able to prevent those collective behaviors from happening when they spread disease. And of course, that ultimate goal begs the fundamental question of how do cells coordinate their behaviors. And something that has emerged as a common feature of collective cell behavior, both in development and also in disease, is the generation and transmission of mechanical forces. So in eukaryotic cells, the main machinery used to generate mechanical forces is the actomyosin cytoskeleton. And this slide here presents a really simplistic overview of actomyosin-based contraction just to make sure that we're all on the same page. So actin is a scaffolding protein that forms filaments. These filaments provide structure to the cell. They're like the poles in a tent. And actin filaments also serve as substrates for a collection of molecular motors. One of those motors is myosin 2. Now, throughout the talk today, I'm going to be speaking about epithelial cells. What are epithelial cells? Anyone knows what they form in our bodies? Yes. Yeah, they line our internal organs, form our skin, they act as barriers, right? So the cells in an epithelium are very closely adherent to each other. They're very closely juxtaposed. Okay, so because I'm going to be speaking about epithelial cells, when I say myosin 2, I mean the non-muscle myosin 2. Many of you may be familiar with actin and myosin in the context of muscle contraction. These are two different genes, non-muscle myosin 2 and muscle myosin 2, but the more we learn about the non-muscle version, the more similarities we find with muscle myosin 2. Okay, so what is this myosin 2? Well, I said it's a molecular motor, and what that means is that it can bind actin filaments and use the energy released from ATP hydrolysis to slide actin filaments with respect to each other. And because these actin filaments are connected to the junctions that hold epithelial cells together, when myosin motors slide actin filaments with respect to each other, they generate a force on the junction. The junction transmits the force to the adjacent cell, and the cytoskeleton and other molecular machines in that adjacent cell can sense that force. And cells can use this transfer of mechanical stimuli to obtain information about what's happening around them and modulate or completely modify their behaviors. Okay, so in our lab, we're interested in how actin, myosin, force generation, and force transmission contribute to the coordination of cell behaviors. And we study those ideas in 3 different models that I'd like to very briefly introduce. The first model is the response to wounds in embryos. So embryos display this striking ability to repair wounds rapidly and to do it with no inflammation or scarring. So in this case, you see the wound there. And they do that through the collective movement of the cells around the wound to repair that lesion. Now, this system is what I would define as our bread and butter. So we've been doing this for the 12 years that the lab has been running. And we love it for a couple of different reasons. The first one is that the signal that triggers the collective behavior, which is the wound, is nicely defined both in time and in space. So that allows us to very easily register experiments at the time of analysis. And the second reason that we really like this system is that it is really easy to manipulate the magnitude of the collective response, so the number of cells that engage in that collective response, as well as the geometry of the response, simply by playing with the size and the shape of the wound. Okay, so really nice system that we really like. However, a couple of years back, and this was at about the time that I turned 40, so you can think about this as a midlife existential crisis, I started wondering, how much of what we learned by punching holes in embryos applies to normal embryonic development. And to alleviate some of that anxiety, we started working on a system where cells move collectively in a developmentally programmed manner. So this system are the early stages of heart development, and so the formation of the heart tube. And what this system allows us to do is compare and contrast mechanisms of damage-induced collective cell movement with mechanisms of developmentally programmed collective cell movement. Okay, you can come in. Now, in these two cases, the cells are moving on the plane of the tissue, so on the XY plane. So we have a third system in the lab, which are these cells here in the center, these big cells, which are a collection of neuronal and glial progenitors. So they form neurons and glia from the central nervous system. And now as I play the movie, you're going to see that the cells will divide very rapidly. And then as time goes by, the cells start disappearing from the surface of the embryo. And the reason they disappear from the surface is that, again, they're going to form neurons and glia. So you don't want your neurons and glia on top of your skin. You want them under your skin. And therefore, before they further differentiate, they need to move inside the embryo. So that internalization happens through the collective movement of the cells perpendicular to the plane of the tissue where they were initially specified, so along the XZ plane rather than movement on the XY plane. So now we can compare and contrast movements on the plane of the tissue with collective movements perpendicular to the plane of the tissue. Okay, now, with the exception of the occasional figure 7, we do all this work in fly embryos. And now we're going to have a problem because you just heard Professor Ruth say that the worm is fantastic. And there is this rivalry between fly people and worm people. So I'm going to tell you that the fly is better. In fact, I'm not going to do that. You're going to tell me why is the fly the superior system to study embryonic development, morphogenesis, collective cell behavior. Any reasons? And I'm not going to give you more than 1 minute. Yes. Excellent. So over 75% of genes involved in human disease are conserved in flies and have phenotypes that mimic what happens in a human. Yes. Exactly. So, embryogenesis lasts 24 hours. We can get, and the reproductive cycle is 10 days, so we can get many, many, many embryos very quickly. So statistical power is not an issue. What else? Yes. Yeah, I mean, developmentally, there's many processes that are very similar in terms of their morphogenesis. For example, I show you formation of the heart tube is very, very similar. Yeah, so there's many reasons. Those are all great. Flies are famous for being genetically tractable, so people have been using flies for over 100 years, and there's collections of mutants and transgenic animals that we have access to. Also, genetic redundancy is not so much of an issue, so you can get rid of a gene, and you're pretty certain to have removed the function of that gene and not to have another one that will take over and compensate for the loss of that gene. We spoke about conservation. They're very cheap. So in my lab, which has a relatively small collection of flies, we keep 800 mutant and transgenic lines with hundreds of individuals for each one of those. And that costs about $5,000 to $6,000 a year. You can talk to any mouse scientist and see if they would be capable of that. And then for those of us who are interested in tissue repair, there are several tissues in the fly that repair and or regenerate. So that's really convenient for us. So many reasons to use fruit flies. And then the other thing that I wanted to highlight is that, as you see in this slide, 99.9% of the work that we do is based on live imaging, so on movies. So we spend quite a bit of time developing image analysis tools to quantify cell behaviors and the coordination of those behaviors from microscopy movies. And you're going to let me use 1 minute for proselytism here. So we put those tools together into an open source software platform that we call Pyjamas, that I encourage everyone to try. And if it doesn't quite do what you want, but you think there's hope, do reach out to us, because we like to collaborate with people to adapt Pyjamas to their needs. The answer is yes. What is the question? Does it do deep learning? The answer is yes. Like everyone else these days, yes, we do have deep learning in there, but actually we have other imaging modalities or other image analysis methods that may be more appropriate for certain images. And what we can do with these methods is automate the detection and tracking of cells, subcellular structures, or supracellular structures from microscopy movies. Okay, now, because you're physics students, I thought it would also be important to highlight that in our lab, we take advantage of computer models, mathematical models, both to generate hypotheses about what's happening in biological systems, but also to interpret the results of the experiments that we do in the fly embryo. And we do that in all three systems, right? So for example, here you have at the top subcellular scale models that we use to understand why is it important to have heterogeneity in the generation of force at the leading edge of these epidermal cells that are moving to close that wound. And then more recently, we developed molecular scale models to understand the dynamics of the cytoskeleton at the edge of wounds as they close. In our work on heart development, we developed cell scale models to understand how periodic cell shape changes could be driving the collective movement of those cardiac progenitors to form the heart tube. And actually, the model predicted that you needed a stiff trailing edge in these cells to rectify those periodic cell shape changes into directional movement. And that's a prediction that we were able to validate experimentally. And lastly, for our neuronal and glial progenitors, we have used computational models to investigate the dynamics of tissue internalization, as well as how the magenta tissue, so the tissue on the sides of those progenitors, facilitates the internalization of those cells by dividing directionally. Okay, so as you see, we address really, really fundamental questions using methods from biophysics, even though we're not biophysicists, and from engineering, as well as from molecular and cellular biology. Now, occasionally, and we don't try to do this at all, we stumble upon translational findings. And I just wanted to finish with that. So we recently asked the question of how does rapamycin affect wound healing? So rapamycin, has anyone heard about rapamycin? You should read about rapamycin. The story of how rapamycin was found is super cool. But rapamycin is a drug that is administered to patients who receive transplants. And the reason that physicians do that is that rapamycin suppresses the immune system and prevents rejection of the transplant. Okay, however, what the surgeon that does the transplant knows is that by giving rapamycin to the patient, they're going to slow down the healing of that wound that they induce when they transplant the tissue. So, you know, it has advantages, but it also has side effects. So we treated our fly embryos with rapamycin, and then we wounded them. And what we found is that in controls, you have the wound that is closing very fast. When we treat an embryo with rapamycin, the wound opens up big and then closes very slowly or completely stalls and doesn't close at all. Now, I'm not going to bore you with the details, but we went on to show that the signaling pathway that rapamycin inhibits, which is mTOR signaling, is actually really important to induce an increase in the volume of the cells adjacent to the wound that facilitates the movement of those cells towards the interior of the wound and rapid wound healing. All right, so I'm going to stop there. All this would not be possible without a bunch of funding agencies, which we're really grateful for. But most importantly, without a group of really, really talented grad students and undergraduates. So Yi here is the person who developed that molecular scale model of cytoskeletal dynamics in wound healing. And they take all the credit if you liked what you saw. If you hated it, you can blame me. Okay, thank you.
Thank you.
Oh. Can I borrow that?
This?
Yeah.
Or the,
Just
oh,
that'll allow me to.
okay.
All right. Thanks again. I think this is super exciting to do this, and good bunch of people here. Hopefully we'll get good questions. So this is just to show that engineers can also turn into physicists. I just added it after, you know, Rodrigo made that joke. So I started in engineering, did electrical engineering, did some bioengineering, worked a bunch outside, and then decided to do a PhD in physics. And I think my background has kind of allowed me, now I think, allows me to do sort of various things in at the boundary of, you know, theory and some experiments and things like that. Okay. So I wanted to highlight something slightly, you know, which has not been brought up in some ways, or brought not that explicitly. I think both the talks did bring this up. Is in biophysics, we deal with something so complicated at the face of it that the big goal is still to find simple descriptions. You know, both of them showed how you can kind of have a very complicated system and still have a simple way of thinking about it. You know, as simple as sort of we write down very simple, you know, equations in physics and things like that, right? So this is my cartoon to show that. You know, this is some old picture by Picasso where he was doing some, you know, I don't know which direction he was going from here to here, but here we want to go from a complicated bull, sort of finding the bare bones that still is enough to explain whatever we want to do, right? So that's sort of the philosophical idea, if you will, in sort of doing theory, at least in biophysics. And this is something very, I'm not going to go deeply into the example, but if you look at this data, it's messy, right? This data comes from blood regeneration happening in monkeys, right? And we won't go into the context of what is getting measured, but it's pretty complicated. But once you know how to really replot this data in a simple way, you actually find that most of this data can be collapsed and replotted in this way, right? There's lots of details going in the middle, but the goal I want to say is this is how it usually looks like. And the goal of our research is basically to find something like this, right? Which then you can model, you can do whatever you want to do with it, right? Of course, it's this simple version is not going to capture everything this thing has, but some aspects of it, right? So that's always also true. Your simple description is not going to catch everything, right? But hopefully it captures what you intended to do at times, right? So in this spirit, I'll give you a few examples of what my group does. But I also had a weird title. I wanted to also push a little bit on saying, well, we can do fundamental work in biophysics. Does it also lead to something further sometimes, you know, as Rodrigo already pointed out, but I'll try to sort of push on that a little bit as well. So broadly in my group, we think about something I was mumbling to some people outside, is structure, function, and dynamics in biology. What I mean by structure function is physicists in particular are very good at structure, right? Can go and look at structure of something and measure it to depth, right? This is what we have been doing for a while, right? Biology brings something interesting which physics doesn't inherently have, function, right? Of course, this table has a function, but the wood, or well, wood has some function in biological context, but a piece of stone or something, you don't think of it as a function, right? You want to understand its properties, you want to characterize it, understand its structure, but the function is it's inherently a biological idea, right? And that is something very new when you think about biophysics that comes in. You have to think about function of what you are studying. So that's what we try to think about, you know, trying to relate these things. And we do this at different levels. This was some work at the level of genomes. This is mitochondrial genomes. So we have mitochondria. They have their own DNA, by the way, separate from the DNA that's in the nucleus. And that DNA is actually quite susceptible to change, much more than our nuclear DNA. So we wanted to look at that dynamics, and we were looking in yeast cells. We think about microbial populations. So you can have a bunch of bacteria and microbes. We know antibiotic resistance is something we all are dealing with. But even at the level of bacteria, they also deal with their own viruses that are constantly killing them. So we wanted to think about their dynamics of how these two populations work with. And this kind of actually gives us a good conceptual model of adaptive immunity in vertebrates. And I'll say a little bit more about it. The third sort of direction my lab has been working for a bit is basically thinking about stem cell populations, right? So in these two cases, I'm allowing DNA to mutate and change. But when we look at a human body, we all came from a single cell, or any multicellular organism, worms and flies included, right? They make different tissues, right? But they have the same genome. So lots of things are happening beyond the genome, right? So DNA doesn't tell you everything. what gets expressed, when it gets expressed, changes a lot of things that you make, right? And how does that kind of non-genetic diversity, if you will, plays into our understanding of many collective biological phenomena, from development to many other things, is something we also think about. But I wanted to go forward and say, well, okay, I should have animated this. Well, so what? Okay, good. So you understand these things. What does it allow you to do more? Can you sort of push forward and ask something more application-oriented questions if you wanted to, just by doing fundamental science? So I just wanted to say that these two don't have to be very separate. At least in biophysics, we are in a place where many things that we study, which may be very fundamental in the sense we are just about now discovering how something works, but it immediately can have an effect on something seemingly very translational. And it's not, depending on if you're interested in that or not. So I wanted to say, some of this work has led us to think about, for example, early detection of mutant clones. I'm going to say a little bit about tumor relapse dynamics and, designing microbial communities, which are important for thinking about gut microbiomes or things like that. So I chose a few projects. So I thought of thinking of, this mutant clone. This relates to some work we did on stem cells on the third column, right? So this was work done with two students, Nika, who was a bioengineering student, and Sophie, who was a physics student. And what they wanted to study, or it started with an experiment Nika was doing, is basically saying, well, if I take a population of skin cells, so this is an amazing discovery, by the way, right? You can take your skin cell and make a stem cell out of it, and from that stem cell, you can do anything. If you take a skin cell of a mouse, you can actually make a whole mouse out of it. Pretty crazy, right? You can reprogram that to a stem cell and then make a whole mouse out of it, right? So clearly somebody got a Nobel Prize for it. And it has really transformed a lot of biological research in a very fundamental way, and also translational research, right? The question we were interested in this context is, well, if you take a bunch of genetically identical cells, they probably should reprogram at the same rate, right? Genetically identical, why would they not reprogram at the same rate? Nika wanted to ask this question, well, maybe there are differences. Can we measure that? And it turns out there are massive differences. Differences are so large that some cells, while being genetically identical to the whole population, can reprogram thousands of times faster. Right? So you can immediately think, oh, this can be very interesting in terms of thinking of how can we make this faster. If we understood how it is, you can immediately start creating populations of cells much, much faster than what we do currently, and in much, much larger scale, because that is a big bottleneck in this problem. But the technology or the ideas, theoretical ideas we developed to look at that data that she was analyzing, we're now trying to use those ideas to actually do a very different thing, which is if you take a bunch of stem cells, so right now you can also extract a bunch of bone marrow stem cells. You can expand them and do something else, right? Some of this come from the very early, when a baby is born, you can actually extract some of the stem cells and you can use them to do anything else in future. But you have to expand them to do something. When you expand them, much like what happens in our body, some of them get mutations, right? And become tumor-like. So if you actually have a culture where you're expanding this thing, you might actually have tumor cells in it, right? And that's why half of these cultures go to waste, right? We don't know how to detect them early enough. By the time we detect it, they're in large numbers. Sort of the same problem we have for tumors most times, right? So we're using the same ideas to kind of now figure out if we can do an early detection of those rapidly growing clones. Because in the other problem, we were looking for these rapidly growing things. And we figured out how to find them. Now we don't want these rapidly growing things, but we know how to find them, right? So you can kind of now turn the problem on its head and use pretty much the same ideas to ask a very different question. Well, this was my schematic of saying, some of these cells really become much more bigger. They contribute much more in terms of number of cells. And you can think of these cells as sort of being, if you think of how many cells each of these individual cells make, if some cells are better than the others, some of them are going to make a ton of them. And if you plot them as a distribution, they'll show up in the tails of these distributions, right? Something becomes much, much bigger than what you expect. So this is interesting in a way, because now if you go back and think, how does a skin cell become a stem cell, by the way, right? It's quite far in some space, right? What is that space you can think of? So if you look at the genes that are getting expressed in a in a skin cell and a stem cell are very different, right? And if not thousands, at least hundreds of genes are completely different in the two cells. If you're really being a physicist and drawing a point in this 100-dimensional space, a stem cell lives here and a skin cell lives there. Right? How does a cell ever make it from one place to the other? Right? We already know, if you do a bit of physics, 3D diffusion is already super hard. Right? You can't find anything in 3D just by randomly walking around. Right? So it's not doing a random search in this 100 dimensions, clearly. Right? At least that was the picture people had earlier. But when we find that some cells can really do this journey so much faster than the others, clearly says that, well, these guys have found some highways to latch onto to reach the other end. Right? They're not doing random walk. Other cells which are not able to do this fast enough probably are stuck around in some random search, right? Because genetically they are identical. So it can't be their intrinsic property. It has to be some early steps they have taken, and some early steps lead some cells to find these highways, and some get lost, right? So now we are looking for these highways, which we wouldn't have otherwise. So I won't go deeply into it, but this led us to thinking about how do you really find these kind of highways, right? Now you're in a very high dimensional space. So this is again a place where physicists can contribute, right? Because we actually have developed, or at least for a while, been thinking about ideas in high dimensional space. If you think statistical physics or thermal physics, those ideas inherently have idea of many, many particles, many, many things moving around. And those ideas from statistical mechanics allow you to think about high dimensional physics, which you can bring to high dimensional biology now, because lots of these measurements are very high dimensional, right? So you actually have frameworks to think about it. And some amount of machine learning always helps. So we've been trying to think about basically how do you infer these trajectories, right? So Spencer Farrell, who's a postdoc right now, she said, well, I can measure a lot of gene expression, or people can measure a lot of gene expression in single cells, but the dynamics cannot really be in this 100 dimensions. I mean, this is now 2000, sometimes 20,000 genes are getting measured, right? So the dynamics cannot be in that high dimensions. Otherwise, we are kind of screwed, right? You really can't do anything. So she said, why don't I compress the dynamics to 10 dimensions and see what I can get away with, right? So what she did was compress the dynamics into 10 dimensions, and she actually learns a dynamical system, much like what Will showed, right? So he was learning a dynamical system where the observable was already low dimensions, right? He already knew he had a phi and an omega, in this case a little bit more. In the other case it was speed and other hidden variables, which he was using Sir Isaac to do. But here she said, well, I don't really know how to formulate this problem. So she used ideas from sort of deep learning, where you can say, well, this dynamical system which is hidden in the middle is low dimensional, but also a neural network, right? So we could actually find interesting ways of thinking about these trajectories. So we can learn these dynamical systems now, right? That make sense a little bit? Okay. So again, this has been useful to think about lung development. So this is a project Spencer is working on right now, trying to figure out how early in fetuses, how do we actually get different types of cells during lung development, and why in certain cases we don't get some, right? So there are lots of developmental defects that happens during lung development. There's a collaborator here in Toronto, so they're trying to look at this, and these kind of techniques are really helping to figure out what kind of trajectories are possible and what can they really do. How much time do I have? 3 more minutes. All right. Sorry? 2 more minutes. All right. Okay. Okay. Let me see what I want to talk about. So I'm going to skip a little bit. I'm going to say, because I want to talk about something we are doing right now. This is all past work, and I think you might enjoy that a little bit. So this is, thinking about tumors and how tumors relapse. So that's a big problem. You give a drug, tumor gets, regresses in our bodies, but then it comes back, right? This coming back, many times it's not genetic at all. It's the same tumor coming back. So how did it manage to hide for that long is something been an important question, and we sort of worked on that with a bunch of people here in Toronto again. And this work has actually led to our collaborators to start a new startup. They have a new way of thinking about tumors because it's not based on genetics of the tumors, it's about epigenetics of the tumors, which is turning out to be much more general than the type of tumor you're studying, right? Because all tumors seem to be hiding in the same way when it comes to drugs. We're now starting to think about liquid biopsies. So now you can go out, you don't have to biopsy the tumor. You can actually just take a little bit of your blood and know how much of the tumor cells you might have in your body, right? This has sort of happened in literally last 5-6 years. But these liquid biopsies are powerful. You can measure in time. And this is what, as physicists, we are great at. We like to make dynamical systems. But in tumors, the problem was you never had dynamical data. You had one biopsy, boom, and then you have to kind of do anything with it. If it's too early, you don't know anything about it. If it's too late, well, it's too late, right? But now you have time series, right? You can try to really think about where the things are going. Can we write predictive models, again, like what Will was showing? If you have time series and lots of them, you can start to make predictions in some ways, right? Okay, I'm going to run through this. Well, we also make some experimental devices to think about microbial communities. I don't think I'll have time to talk about it. And again, this is so a student, Wei Yu, who developed these things, is now actually working for a startup in Chicago, trying to make these devices where they really want to evolve these microbial communities, right? So many of these things which are really fundamental in the sense we were measuring stuff people don't measure right now. That was the goal. But then once you're able to measure something new, you can really do something new in a way which you might not have intended your system to do, right? So there's a lot of fundamental work then leads into this immediate, translational or immediately applicable stuff, which I think is exciting about biophysics compared to some of the other things. Okay, this is sort of the phage bacteria stuff, which is sort of like a Red Queen hypothesis, if phages are running, bacteria is running behind it. Could be true for any viruses. And our simple models kind of give us stuff which you see in flu, right? So if you look at flu phylogeny, you actually see this running, from time to time. I'm going to close in like 30 seconds, okay? So even in these very simple conceptual models, you can see what happens in real data. And, now you can kind of use this kind of data to this kind of conceptual thing to understand what real data is doing. Finally, we've been starting to think about whole organisms. And I think the place which has been exciting is, of course, these guys have been thinking about whole organisms. I was thinking just about populations of cells so far. But regeneration seems to be an interesting place because it's a very physical process, right? You chop the head off of an organism and it comes back, right? So there's a lot of physics going on. And it's not very developmental in the sense that it's not starting from a single cell doing something very stereotypic, right? You could chop off in this way, you could chop off in this, it's getting a bit morbid, but you see what I mean, right? You can take a little slice of, you can take these worms, the worms we are thinking about, starting with flatworms. In these experiments we can do, it's a very simple experiment. You can just, chop them off in any which way and see how they become a full worm again, right? So now it's kind of crazy. You can take this part, you can take the tail part, you can chop it off in this way. Any which way you chop it off, it becomes a whole worm again, right? It's a crazy kind of setup, yeah. Both half becomes a new one. It's a totally Deadpool thing. And there's lots of them now, right? So it's a bit crazy. So yeah, how does that happen? It's not very developmental in the sense that the initial condition is so different, right? You can take any piece, so it has to kind of problem solve its way to finding this thing, right? And so these are the kind of problems where I think, kind of frameworks we have in physics may be useful. Okay, I think this will stop. Yeah, I've mentioned most of the people who worked on this. No picture. Okay, thank you.